Welcome everyone. Today we are going to learn about resting potential of neuron. But before that, I want to briefly recapitulate what we have learned in my previous video. That is, the resting potential of neurons. The value differs between vary between -40 to -90 millivolt in different species. Now, why there is a resting membrane potential of neurons? Because of the movement of ions. Now, why there is a movement of ions? Because of the presence of ATP-driven pump. So this is the first trigger that actually helps in the generation of resting potential. Now, ATP-driven pump, there are many different ATP-driven pumps present that actually generate ion concentration gradient. Ion concentration gradient means there is a concentration difference in the inside and outside of cell. Now, I have given here an example of sodium-potassium pump. So, as you can see, this pump exports sodium out of the cell and import potassium. So, the sodium concentration exterior is high than interior. The potassium concentration exterior is low than inside of the cell, and there is different ATP-driven pump present for different ions. I have not mentioned here. There is also a different ATP-driven pump present that actually generate the chlorine concentration. Now, why I am mentioning these three ions? Because it has been found that membrane potential is actually generated contributed by these three ions: sodium, potassium, and chlorine. So chlorine concentration is also different. As you can see, the exterior is higher and interior is lower. Now, because there is a difference in the concentration of ions inside and outside, this is the chemical force. Because there is a non-gated channels. These channels are actually responsible for the generation of resting potentials. As I have mentioned, ion movement is actually responsible for the generation of resting potential. So ions channels actually helps in the movement of ions. Remember this. So this is the driving force. As you can see, I have actually drawn the movement because through the channels, ions move from higher to lower concentrations. As you can see, sodium concentration is higher outside, so it will move from outside to the inside. And because the potassium concentration is higher inside, so the potassium will move from inside to the outside, and chlorine will move from exterior or outside to the inside. So this is the movement of the ions, and that movement is actually generated by the ATP-driven pump, because they break ATP to perform these functions, import and export, and they generate difference in the concentration that is called ion concentration gradient or the chemical force. Now, non-gated ion channels actually utilize this concentration difference to move the ions, and now the second point comes that because these ions carry a charge, so the movement of ions will also create a difference in voltage. We call voltage gradient. Like the ion concentration gradient, there is a voltage gradient created. That means the inside and outside of the cell has a difference in voltage. Just as I mentioned, that outside is actually more positive charge present than inside. So this is also called membrane potential or electric potential, or you can say voltage gradient. Now this is the electric force. Now these two forces actually just oppose each other. Ion concentration gradient or chemical force try to move the ions, and this membrane potential or electric potential or voltage gradient try to stop the movement of ions. This is called electric force. Now together we call it electrochemical gradient. And now when this membrane potential try to stop the movement of ions, the value of these two force becomes zero. Remember this. So the membrane potential at which the value of electrochemical gradient becomes zero, that means at which there is no net movement of ions. This is called equilibrium potential, or E specifically. Now, if potassium ions is responsible for the generation of resting membrane potential, the equilibrium potential will call it EK. If sodium is responsible, then a potential will be generated that will be ENA. That means the equilibrium potential for sodium. And there is a particular, if chlorine channel is responsible for generation of resting potential, then we will call it ECl. Because why? Because if any channel is open for a fraction of milliseconds, this potential will be generated, equilibrium potential, particularly for potassium if it is potassium channel, for sodium if it is sodium channel, or chlorine or if it is chlorine channel. This phenomenon actually discovered by Alan Hodgkin and Bernard Kazt in 1949. They took the advantage of squid giant neuron and they measured the value of resting potential. They got a value is approximately minus 70 millivolts. So they got a value of minus 70 millivolt. And in general situations, we also find that animal cells actually has a resting potentials of minus 70 millivolt. Now, Alan Hodgkin and Goldman-Hodgkin-Kazt try to understand which ion is actually responsible for the generation of resting potential. Okay, so what they did, they performed a very simple experiment. They changed the concentration of these three ions exterior. Okay, now when they changed the concentration of sodium and chlorine, they found that little value of this membrane potential changes. That means they are not very correlated with the membrane potential. Now when they change the concentration of potassium outside, they find that this value is actually changing proportionately or linearly. That means potassium ions is important for the generation of resting membrane potential. Now there is an equation by which we can actually validate the value of this equilibrium potential, that is called Nernst equations. Through Nernst equation, we can also derive the equilibrium potential. That means the membrane potential at which there is no net movements, because this is the fundamental phenomena of these channels, that for a few milliseconds when it opens, as because through the channels ions move very fast, 10 to the power 9 ions per second. So as because very fast these ions will move, then what will happen, a membrane potential will be, or voltage gradient will be generated at that point. Then equilibrium gradient or the movement of ions will stop, and this is called equilibrium potential or resting potential. Now we know that through this experiment, we know that potassium channel is open. Many potassium channels actually remain open, and very few sodium and chlorine channel is open. Now we will also validate this value with Nernst equations. Let me show you how Nernst equation is derived. As you can see, there is a delta G for C. Now delta G for C is free energy change for the chemical force, that means the ion concentration gradient, and delta G for E means the free energy change for the electric force, that means the membrane potential that is generated. Now for the delta G, the formula is minus RT ln the ion concentration that is outside divided by the ion concentration inside. And for the delta GE, there is a E, that is the electric potential or the equilibrium potential, you can say. Z is the valency and F is the Faraday constant. Now that becomes 0 because at certain conditions the two actually force just balance each other or equilibrates. That's why the value becomes 0. Now if we exchange this value, the minus RTLN becomes positive, and if we divide this ZF, we get the value that is the equilibrium potentials. Now as I have mentioned, equilibrium potential is very important because for a few milliseconds, just a brief time of periods of time, if the channel is open, through that opening channel, when ions move, they actually generate this equilibrium potential. At that potential, the further net movement of ions will stop. That's why this value is important. Now the R is the gas constant, the value is 2. T is the absolute temperature at 37 degrees centigrade, body temperature I am taking here. We have to add 273 more to get the degree Kelvin. So it is 310 degree Kelvin. So I have write here 310, and 1 is the valency of the ions, as because I have taken example of potassium, sodium and chlorine, so it is a positive 1 or 1 only, and the Faraday constant is 2362. Now the whole value, now here the natural logarithm, if you convert it log, it gets the value 2.3. Now if you multiply all this and divide this, you will get 0.61, that is log in ions of outside concentration, inside concentration. Now the potassium, the equilibrium potential for the potassium will be 0.61 log, as because outside here concentration is 5 and 155, you will get a value of minus 98 millivolt. For the sodium equilibrium potential, that means if sodium channel is open and if sodium channel is responsible for the generation of resting potential, then ENA or equilibrium potential or the membrane potential will be generated. That will be as because their outside is higher concentration, it will be positive 61 millivolt. And in case of chlorine, as because chlorine carries negative charge, so we will convert it and it will make a difference, that is in this case outside concentration is divided by inside, so we will convert it into inside divided by outside, as because it is negatively charged, so it will get generalized. Now that's value also get approximately 61 millivolt. I have taken the inside value of the chlorine is 11 because it can vary from 4 to 30, so I have taken this example as to actually simplify this value. So what we have found here that if sodium channel is open, then what will happen? After few milliseconds, this potential will be generated, and if Allen-Hodgkin and Barnard-Kazt measured the neurons, they will got a positive value, but they do not got a positive value, they got a negative value. So this is not true. Sodium ion channels are not open, and this is equal for chlorine, because through the Nernst equation we have also got a value of equilibrium potential of the chlorine that is positive in value. So this is not also true. But what we are getting through the equilibrium potential of potassium, a negative value. But remember, Allen-Hodgkin and Barnard-Kazt got a value of minus 70 millivolt, and we know that in general the animal cells also has resting potential. That value is approximate minus 70 millivolt, or it varies from minus 40 to minus 90 millivolt. Now, why this value minus 98 millivolt is not exactly the same value that actually derived by the experimentations or we got in real physiological conditions. The reason is, as because I have mentioned that many potassium channels open, so potassium potential is actually the main potential generated or equilibrium potential, but few sodium channels is open, few chlorine channels open, through which few sodium will enter, few chlorine will enter, and that will actually change the value from minus 98 millivolt to 70 millivolt. Now there is another equation that's called Goldman equations. Remember this Goldman equation. Now not only Goldman but Allen-Hodgkin and Barnard-Kazt, they also help to derive one equations that's called Goldman equations. In that equations, they taken the help of, or they taken a value that is called permeability. Okay, permeability of ions. Now why they have taken the example of permeability? Because they emphasize that if for the generation of membrane potential or resting potential, whatever it may be, for the generation of potential, membrane potential, if multiple ions is responsible, then we have to take the extra value, that is permeability of the ions, along with the ion concentration. Remember, Nernst equation is for a particular ions like EK, ENA or ECl. That means it only derives the value of a single ion channels that generate the membrane potential. But in physiological conditions, what we have found that many potassium channels actually remain open, few sodium is actually open, few chlorine is open. Now concentration remains same, but the permeability of the neurons is different, because the potassium permeability is higher in resting conditions and very low for sodium and very low for chlorine. So we will take the permeability value also, and that equation is called Goldman equations or Goldman-Hodgkin-Kazt equation. Hope this video will help you. If you have any doubt about this topic, you can write me in the comment section. Thank you for watching, and always keep learning.